Hi guys, in this lecture we are going to talk about the biomolecules. So what are your biomolecules? Simply the biomolecules are the carbon containing compound which is found in the living tissue or the cells, right? So carbon containing compounds, molecules you can say, or compounds. Okay, so based on the weight we just classify the biomolecules into two categories. The first category we have is your micromolecule. Second category is your macromolecule. So micromolecule are those molecules which got the weight between 18 to 800 dalton, whereas macromolecule having the weight more than what 1000 dalton generally. Okay, so the examples of the macromolecules are your amino acid, nitrogenous bases, or the adenine, guanine, thymine, and all the fatty acids, sugar, etc. So these are what your micromolecule. And what are your macromolecule? These are what your carbohydrates, lipids, proteins, and then the nucleic acids. So actually, what is happening? Your micromolecule, they are combining together, right? They're coming together, and what giving you macromolecules. So micromolecules can be termed as what the monomers, and macromolecules can be termed as what the polymers. So in this lecture we are going to talk about each and everything in details. So let's start with the carbohydrate. Okay, so carbohydrate is basically what these are the substances which is what having the carbon, hydrogen, and oxygen atoms. Or in other term, we can simply say these are the hydrates of the carbon. And if I talk about the general formula of the carbohydrate, so this is going to be the CXH2O and Y. So this is what the general formula of the carbohydrate. We classify the carbohydrate into three categories. The first one is your monosaccharide, second one is your disaccharide, and then the third one is your polysaccharide. Mono simply means what? One, and saccharide simply means what? Sugar. One sugar, right? And then we have a disaccharide. Di means two, and saccharide simply means what? Sugar. So we got the two sugar. And polysaccharide means, poly means again many, and saccharide simply means what? Mini sugar. So in monosaccharide, we got the simple one sugar molecule. And if what two sugar molecule they come together, that is going to be your disaccharide. And in the polysaccharide, we have the many sugar molecules. So carbohydrate are simply what known as what? Sugar. So one sugar, monosaccharide, more than one, and we accordingly we have the name di and poly. Okay, what are monosaccharides? So again, as I told you, simplest carbohydrate. And the example here is your glucose, the very, very famous one, fructose, and then the galactose. The general formula is C6H12O6. Okay, so this is the structure of the carbohydrate. Which one? The glucose. Okay, so this is the structure of the glucose. Now let's talk about the disaccharide. Di simply means what? Two sugar molecule. So in the disaccharide, if two or more than two monosaccharides they join together, then you will get the disaccharides. And in this case, what's happening? A bond is formed. That bond is known as what? Glycosidic bond. So see, if this is one glucose molecule, right, and this is the another glucose molecule, both the molecules they both are joined, and that bond is basically known as what? Glycosidic bond, right? Here in this joining process, there is a reaction known as what? Condensation reaction. Condensation reaction is going on, and in this condensation reaction we get a water molecule, right? So joining of two sugar molecule or of two glucose molecule, that reaction is condensation reaction, and we get the water molecule, and the bond between these two sugar molecule is your glycosidic bond. So as you can see here, the joining of the two monosaccharide, water molecule is being released, and then the reaction is known as what? Condensation reaction. So here we see the example of the monosaccharide. So in the previous slide we just saw the glucose is a monosaccharide, galactose is a monosaccharide, fructose is a monosaccharide. So here we are talking about the disaccharide, means we are going to join the two monosaccharide. So two glucose, if they are joined together, we'll get the maltose, which is a disaccharide. If glucose and galactose, the two monosaccharides which are joined together, and that is known as what? The lactose, that is what? Your disaccharide. So glucose plus fructose, which is what they are going to join together, and we'll get a disaccharide known as what? Sucrose. It's important, you have to know this. Okay, so in this case you see the structure of the glucose. So as glucose is a 6 carbon compound, right? So this is going to be your first carbon, second carbon, third carbon, fourth carbon, and fifth carbon, and to this fifth carbon you have the sixth carbon. So this is what C6H12O6. Okay, fructose and then the galactose. So these are the monosaccharides, and all the sugar molecule, all the carbohydrates, they are formed by simply just joining either the glucose chain or the glucose fructose chain or glucose galactose chain. All right, so we have just talked about all those monosaccharides and the disaccharides. Okay, now what is alpha glucose and what is beta glucose? Right, simply if you see this structure of the glucose, so this is the carbon number 1, carbon number 2, third carbon, fourth carbon, and to this fifth carbon you have the sixth carbon attached. Right, so specifically if I just ask you to just look this first carbon, here we have the hydrogen and then we have the what, OH group attached. So if to this first carbon, if the hydrogen is above the carbon, so that kind of a glucose molecule is basically known as what, alpha glucose. If the hydrogen is below, if the hydrogen is below the plane, that kind of a glucose molecule is basically known as what, beta glucose. Right, so simply if the hydrogen is above the plane, alpha glucose, hydrogen is below the plane, and that's what your beta glucose. We are talking about this with respect to the carbon number 1. Okay, now see, here we are talking about the how the maltose is formed. So maltose is simply what, glucose plus glucose, right? So maltose is a disaccharide and it is formed by two monosaccharides. So that is your glucose plus glucose. So this is the glucose structure. This is the glucose structure. This is going to be your carbon number 1. This is your carbon number 1, 2, 3, 4, and this is your 5 and 6. So same case, 2, 3, 4, 5, 6. So here what is happening? The bond is formed between the carbon number 1 and carbon number 4. Specifically, the OH and OH, they both are going to participate in the what, condensation reaction. So OH of this and H of this, they are going to what form, and water is going to be released, and then the bond is formed. That this bond is basically known as what, glycosidic bond. So glycosidic bond is found in the carbohydrate, and it's what, formed by the condensation reaction where we get the water molecule released. All right, what kind of the glucose is this? Again, this is the alpha glucose. This is the alpha glucose. How we have come to know? Because of this hydrogen. Hydrogen is above the plane. Okay, here we can see how the sucrose is formed. Okay, so simply as you can see, the same thing is going to happen here. What is happening? The carbon number 1 and then carbon number 1, 2, 3, 4, fourth carbon. So this is going to what, interact, and the OH and H, water will be released and the bond will be formed. This is known as what, glycosidic bond. Okay, so what are the function of the monosaccharide and disaccharide? Simply, so monosaccharide and disaccharides, they are going to what, the simplest sugar or the simple sugar. So this is going to be the good source of the living organism, right? As they are the simple ones, so they are easily soluble, right? And hence, if it's easily soluble, this simply means what, they can be transported throughout the organism's body. So if I talk about the animals, glucose is transported in the dissolved plasma state, and in the plants, sucrose is transported in the phloem sap. Okay, now let's talk about the polysaccharides. Polysaccharides simply what, many saccharide, many sugar molecules, they are what, joined together. So polysaccharides are your what, macromolecule, which contains what, hundreds and thousands of the sugar molecule. They are joined together and they are what, giving you the polysaccharides. As this polysaccharide is a complex molecule, so it generally do not dissolve in the water. Okay, this polysaccharide is what, good for the storing purpose. So we just talked about the monosaccharide and disaccharide as they were the simpler one. So that what I mean, that can be used for the transportation purpose. But here the polysaccharide is what, the complex molecule. So we are going to use this for the storing purpose. So the very famous example is the starch and then the glycogen. Right, so glycogen, which is generally found, which is found only in the animals, and this, this glycogen basically means what, the exoskeleton of the arthropods. And you'll be surprised to know that the glycogen in the animal world, right, this is going to be what, most abundant carbohydrate, right? So glycogen is going to be our what, most abundant carbohydrate. And if I just talk about the plants, right? So in the plants, what is going to be? It is going to be your cellulose. So this is the cellulose, right? So plants, cellulose is going to be the most abundant carbohydrate, right? Okay, so we got the starch also. So starch is found in the plants, so glycogen is found in the animals, and this is what the most abundant carbohydrate in the animal. And starch, which is found in the plants, we got the two forms of the starch, amylose and the amylopectin. We'll talk about it. I mean, what kind of a chain is there, what kind of a linkage is there, we'll talk about this. We have a chitin also, which is what found in the animals, and this can also be known as what animal starch, which is what stored in the liver or the skeletal muscle. Cellulose, the very famous, which what makes the cell wall. Okay, so if you look the structure of this is the glycogen molecule structure, and as I told you, glycogen is what most abundant the carbs in the case of the animal. So here, what kind of a linkage you see? See, this is the what glucose molecule, right? And this is your carbon number 1, carbon number 2, this is carbon number 3, carbon number 4, carbon number 5, and to this you will have the carbon number 6 attached. Now here, between the carbon number 4 and carbon number 1, we have a bond, that is what known as what alpha 1,4 linkage, alpha 1,4 glycosidic bond. So this can also be known as what alpha 1,4 glycosidic bond or the glycosidic linkage. Why I'm saying alpha? Because see here, it is going to be something like this. Not OH, the H will be above, so that's why it is going to be your CH, and here it is going to be OH. C, here it is going to be H, and here it is going to be OH, right? So that's why the alpha glucose, right? Okay, what you see here, the 1,6 linkage. 1,6 linkage simply means what? This is going to be your 6th carbon. Okay, this is the first carbon, 2nd carbon, 3rd carbon, 4th carbon, and to this 5th carbon you have the another carbon attached, that is your 6th carbon. And here if you'll see, this is the what? First one. So right, so alpha 1,6, so this is what the bond between this one. So that's why what your 1,6 linkage. This is again what kind of a bond? Glycosidic bond. So generally, if you have the if you have the multiple sugar molecule, right? So this is the one sugar molecule, another. So if you have the one chain of the sugar molecule, right? So what kind of a bond you will find? You'll find the alpha 1,4 glycosidic bond. So this is the general case. But when you have the what branching, so in this case, if any kind of a branching is there, so here you will find the 1,6 glycosidic bond, 1,6 glycosidic bond. Please remember this. In the case of the branching, you'll have the 1,6 glycosidic bond, and in the case of the linear chain, you'll have the 1,4 glycosidic bond. Okay. Now, if we'll talk about this in plants, the storage polysaccharide is your starch, right? So starch is what? The storage polysaccharide, because it is going to be complex one. And starch is a mixture of what? Two substances, that is the amylose and amylopectin. What is amylose and amylopectin? See, amylose is a long chain of the alpha glucose, and here you'll find the alpha 1,4 linkages. Simply means what? In the case of the amylose, the bonds are what? Alpha 1,4 glycosidic bond. This simply means what? This is going to be what? Something like this. But not only this one. In the case of the amylose, we have something else also. In the amylose, the structure, there is what another kind of a bonds formation there, that is the hydrogen bond formation between the molecules. So this lead to what? The formation of something spiral kind of a structure, and this makes the amylose what? Somehow more complex. In the amylopectin, what we see the alpha 1,4 linkage and the 1,6 linkage. This simply means what? Amylopectin is what? Kind of a branched structure, right? So amylopectin is a branched one, and amylose is simply a linear one. But yeah, we have the additional the hydrogen bond is there, and that's what making it a kind of a spiral structure, right? Okay, so as you can see, this amylose contain the alpha 1,4 linked glucose, and the glucose molecule in all chain have the same orientation. So alpha 1,4 glycosidic bond in the amylose. So structure polysaccharide, the one which gives the structure, so that's your cellulose. And in this cellulose, the glucose molecule which is present in the beta form. Alpha form simply means what? This is the sugar molecule, right? And this is the sugar molecule, this is the carbon, and this is the H, and this is the OH. So this is going to be your alpha. In the case of the beta, we have the just reverse. The structure is got reversed, and in that case, we'll see the beta form. Okay, so alpha beta form I think it's clear to you, right? So this simply means that the adjacent glucose molecule in the chain are upside down, right? That's what I just told you in relation to the one another. Okay, fine. So as you can see in this image, what they are telling you, yeah. So this is what beta 1,4 glycosidic bond. How? See, this is structure. This is what? This is your H here. So this simply means what? Beta. Again, you have the H here. So this is what? Your beta. Means simply means you must be having OH here, must be having OH here, and the both OH and one H, they what? They just interacted, and the water molecule is what? Is being released here. So that's what your beta 1,4 glycosidic bond. Beta 1,4 means what? One carbon, first carbon of this and fourth carbon of this, another sugar molecule, they are what? Coming together, and that's what you're giving you the beta 1,4 glycosidic bond. So cellulose contain what? Beta 1,4 what? Glucose. And you'll be surprised to know, I mean, along with this beta 1,4 linked molecule, we have the hydrogen bond. That's what I told you now. These are what? Are the hydrogen bond, and this is what? Form the microfibrils. So this is giving the extra toughness, right? Okay, so what are the tests for the carbohydrate? The test for the carbohydrate is basically what? Based on the test that we basically use, we have the reducing sugar and the non-reducing sugar. The reducing sugar are basically those sugar which are able to reduce the Bendix reagent. Okay, so suppose you just take any sample, and along with this reagent, that is the Bendix reagent, and you just heat it, right? You're just going to heat it, and then this reagent got the specific color, right? Let's suppose the blue color, right? So reagent got blue color. So the sample we just taken is your carbohydrate, and that carbohydrate, let's suppose I've taken the glucose molecule. So let's suppose this is the monosaccharide. So monosaccharide, and along with this reagent, Bendix reagent, which is blue in color, and you are going to heat it. If there is a change in color, so orange, red color kind of a precipitate is there, red kind of a precipitate is there. This simply means what? This sugar is your reducing sugar. So that kind of a sugar which is able to reduce the color of the reagent, that's what your the reducing sugar. The examples are basically what? The small molecules, all those, the monosaccharides and the disaccharides, right? So these are the examples. Some of the disaccharides are what? Acting as a reducing sugar. So all monosaccharides and some disaccharides, they are what? Acting as a reducing agent, and they are going to what? Reduce the blue Bendix solution to the orange red precipitate, and that's what? The reducing sugar. So we do that, such kind of a testing to for the, I mean, to check the carbohydrate. Non-reducing sugar, simply if you go for the reducing sugar test, if you fail to do so, this simply means what? That is going to be your non-reducing sugar, and the very famous example is your sucrose. So sucrose is what? Your non-reducing sugar. Okay, and some molecule along with this, we have a starch. So a starch also don't go with the Bendix reagent test. So we have the another thing, that is what? We take the iodine, and that along with this we have the potassium iodide solution. So iodine plus potassium iodide solution, and we just put few drops of it in the starch containing what, let's say potato or anything starch containing molecule or the substance, and what you'll see, if there is a change in the blue black color, so if there is change in that color, so this simply means what? A specific color is your blue black color. So this simply means what? That yeah, this molecule or this fruit or this vegetable contains what? A starch. Okay, so iodine plus potassium iodide solution is what? Few drops of it is what? Pour on the starch containing molecule or the fruits or the vegetables, and if there is change in the blue black color, this simply not give the confirmation that yeah, this molecule or this item contains the starch. Okay, so we just completed the carbohydrate. Now it's time to discuss the lipids. So what are lipids? Simply, this is going to be the diverse class of the organic compound, and this includes your fats, oils, waxes, and then the steroids. Okay, so it's what? A diverse class, having the fats also, the oils, waxes, steroids also, right? Just like the carbohydrate were having the carbon, hydrogen, oxygen also, but here in the case of the lipids, you have all these carbon, hydrogen, oxygen also, but here the oxygen is what? Present in the very small proportion. And if we talk about the lipids, the examples of the lipids, we have the triglycerides and then the phospholipids. So these are the what? Lipids include what? Triglycerides and the phospholipids, and these lipids are insoluble in the water, but yeah, they are soluble in the alcohol, you can say ethanol. Okay, triglycerides. Let's talk about the triglyceride. In the triglycerides, we have two things. The first thing is your glycerol, and then the second thing is your fatty acid. So this is the structure of the glycerol, where we have the 3 carbon molecule, and to which we have the hydrogen and oxygen attached, right? And this is the what the fatty acid. What is fatty acid? Simply long chain of the carbon. Okay, and at one end, let's suppose this is the one end of it. So at one end you will have the carboxylic acid, and the rest of the position will be filled by the your hydrogen, hydrogen, hydrogen. So this is what, that's what the basic definition of the fatty acid: long chain of the carbon, carbon, hydrogen, and at one end you have the carboxylic acid. That's what your fatty acid is. So this OH that you see, to this OH you have the one fatty acid attached, to this OH another kind of fatty acid attached, to this and you have the another kind of fatty acid attached, and ultimately if you are attaching 3 fatty acid to this glycerol, you will get the triglyceride. 3 glycerol molecule are what, or the 3 OH hydroxyl group of the glycerol is what involved in the what the bond formation with the fatty acid. Again here you'll see there is a what condensation reaction going on, and you will get the water molecule. The bond between the glycerol and the fatty acid is basically known as what, ester bond, ester bond. Okay, so what is what, triglycerides. So in the triglyceride, the backbone of the glycerol is going to be the backbone, and with this the 3 fatty acid molecule is what going to attach, and the bond is what, your ester bond. Okay, so what is the function of the triglyceride? Simply help in the insulation, okay, buoyancy, and it's acting as an energy reserve. So like the adipose tissue. So adipose tissue stores the what, the fat. So these are what, in the form of the triglycerides. Okay, provide insulation, like suppose all those the mammals like the whale or the dolphin, they are there in the colder areas, so their body is insulated just because they have the fatty acids, right? That triglyceride specifically. So in this image what you see, the fatty acid molecule, so this is the long chain of the carbon, hydrogen, and at one end you have the carboxylic acid, and this is the glycerol molecule. So glycerol molecule, you have the OH, 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 and the rest position is filled by the hydrogen, and the what, the OH, and here the OH. So the condensation reaction will go on, and you get the water molecule, and hence one ester bond is what, one. Okay, so again the glycerol plus 3 fatty acid, you'll get the triglycerides. And yeah, so what is fatty acid? We just discussed is a carboxylic acid with the aliphatic chain, and this fatty acid could be the saturated or the unsaturated one. Okay, saturated simply means what, the what, no double bonds. The saturated fatty acid will have the no double bond. Simply means what, long chain, long chain. At one end you have the carboxylic acid, and rest position is being filled by the hydrogen. That's what your saturated fatty acids. Okay, saturated fatty acid. And if any molecule, if any long chain, yeah, long chain is there, but if you have the double bond, one or more double bond, that kind of a fatty acid can be termed as what, unsaturated fatty acid, as you can see in this image. So this is the long chain of the fatty acid, and at one place you see the double bond, so that's what your unsaturated fatty acid. Okay, so if you have one or more double bond in a long chain of the fatty acid, that's what your unsaturated fatty acid. So what is the difference between the saturated and the unsaturated one? So saturated contains single bond, that's good, and these are what consumed, not to be consumed more than 10% of the total calories if you are taking. So it is what consumed in a very, very less proportion. And if you are taking in a high proportion, this leads to the heart related disease. Okay, generally or the saturated fatty acids are what got the high melting point, and they are what, solid at the room temperature. So at the room temperature you'll see the saturated fatty acid in a what, solid state. And this saturated fatty acid compound, they do not what, spoil quickly, right? So the examples include what, your cheese, your butter, your the coconut oil, your vegetable oil, your peanuts, your meat, the fried foods, and all, right? And when we talk about the unsaturated fatty acid, right, so contain at least what, one double bond, and not to be consumed more than 30% of the total calories per day. Okay, so good for consumption, but excessive may increase the cholesterol. So if you are taking it, that's fine, but if you are taking it in a high proportion, so it lead to the what, cholesterol level, high cholesterol level. But yeah, if we talk about the state, so generally they are liquid state in the room temperature, and as they have the low melting point, and they got spoiled quickly. Okay, so the food sources, if you just talk about the walnut, flax, sunflower oil, soybean oil, fish oil, these are what the source of the unsaturated fatty acid. All right. Okay, now let's talk about the phospholipids. We just talk about the triglycerides having the glycerol and 3 fatty acid. Now let's talk about the phospholipids. So phospholipids are basically what, just like the triglyceride, in which one fatty acid is replaced by the phosphate group. So as you can see here, this is what the glycerol molecule, right? This is the glycerol molecule, right? This is the one fatty acid, this is what your fatty acid 1, and this is your fatty acid 2. But the at the third place you have the phosphate group attached, right? So that's what your the phospholipids, how they are made. These are the amphipathic molecule having both the what hydrophobic group as well as the hydrophilic group. So if you look at the structure of this, so this is the what basic structure of the what fatty acid, having the head part, this is the head part, and this is the tail part. So tail part are your fatty acids, so these are your fatty acid, and they are what hydrophobic tails. Okay, as they are fatty acids, so they are what hydrophobic tails. And then the tail part, we have the phosphate group is present, so this is what hydrophilic, means it loves the water, right? So generally, if you'll have the phospholipids something like this, we just like it's there in the any medium. So by on their own, they'll what form the micelles. They'll form the what micelles. So what head part towards the water surface, and the tail parts towards, it will what try to avoid the water surface. So what such kind of a structure is what your micelles. All right, micelles. Yeah, something like this. So micelles. Okay, and the very basic the function of the phospholipids that you see is what the cell membrane. The cell membranes are what they are made of the phospholipids, right? So these are the what, these are your phospholipids. And if you to talk about the cell membrane, along with the phospholipids, you have the cholesterol, you have the carbohydrate molecule, you have the proteins, intrinsic protein, extrinsic protein, all these are there. But yeah, the main composition is the phospholipid, right? It's very huge in number. So lipid bilayer, the phospholipid bilayer is what there in the cell membrane. Okay, so this is again the structure of the phospholipid. Okay, having the hydrophilic head, means it loves the water. Okay, loves water. And hydrophobic tail means it hates water. Why? Because it got the what fatty acid tail. Okay, so as you can see again, the cell membrane structure. So inside and outside of the cell, you got some protein, intrinsic, extrinsic protein. And if you just see the cell membrane, you have the phospholipid bilayer. Okay, so test for lipid, what are the tests? Simply, if you have if you want to check for any substance, so just mix the substance with the absolute alcohol, and then just decant the ethanol into the water. And if you get some kind of a milky emulsion, this simply means what you have the lipid. Okay, what are steroids? Simply, I mean, it's again the example of the what your lipid, right? So lipid, what it's what example of your steroid is example of the lipid, right? So lipid that do not have the general structure of as the other lipids. So that's what your steroids are. So this is the basic carbon skeleton of the steroid. And if you are attaching any specific group to this, so this is the specific group if you're attaching, this can be termed as the what cholesterol. So the examples of the steroids are your cholesterol, the testosterone, estrogen, the progesterone. All these are what your these steroids. Some steroids are basically your hormones and have the what and therefore lipids have the endocrine function. So the function of the steroid and the steroid hormone could be your carbohydrate regulation, mineral balance, reproductive function, inflammatory response, the stress response, and then the bone metabolism, right? So these are the what the steroid related hormone function. So simply those lipids which you do not have the specific structure just like the other lipids, but yeah, it's kind of what other than the different one. So this is the basic structure of the what the steroids, you can say. And if there is what modification, if there is an addition of this, if any, let's say if you are adding a CH3 group or any specific group you are attaching, based on this you get this specific steroid molecule. So mostly these see the steroids are the hormones. I hope you like the lecture. If you have any kind of a doubt, you can just ask me. Thank you.